Welcome to the deep dive. Our mission here is, well, it's to unpack complex topics, really getting into the source material so you walk away with the insights and clarity of an expert. And today we're plunging into a field that honestly sounds like pure science fiction, but it's rapidly becoming a tangible, life-changing reality. We're talking about regenerative medicine. So let's unpack this right away. What is regenerative medicine really at its core? It's an incredibly fascinating mix, actually, a blend of several distinct scientific disciplines. We're talking cellular and molecular biology, sophisticated tissue engineering, and cutting-edge biomaterial science, all working together with this one powerful goal, designing therapies that can either restore lost function or completely maintain or repair cells, tissues, even whole organs damaged by disease, injury, or just, well, life. It's really interesting if you think about the natural world. Many other complex organisms out there, like think of a salamander regrowing a whole lost limb, or a starfish just effortlessly regenerating an arm. They keep this incredible capacity for true regeneration right through their adult life. But humans, well, we seem to have made a kind of evolutionary trade-off. We prioritized speed and strength of repair, like how fast a cut closes to stop infection. But that definitely comes at a cost, doesn't it? And that cost is often scarring, which can lead to a pretty significant loss of function, maybe reduced mobility, and it certainly can impact how things look, the aesthetic appeal. We repair fast, but often imperfectly.
And what's truly fascinating here is precisely how regenerative medicine is trying to overcome that exact evolutionary trade-off. It's really about bridging that gap, between our rapid, maybe scar-forming repair and the ideal of true seamless regeneration. The progress, just over the last decade, particularly in fundamental stem cell biology and crucially in cell-based regenerative medicine, it's been absolutely immense. And when we connect this to the bigger picture, it really promises to address critical limitations we currently face in traditional medicine. Think about the chronic global organ shortages, right? So many patients waiting. Or the morbidity, the problems associated with donor sites when tissue is harvested from patients for grafts. And of course, the persistent challenge of immune rejection, which forces lifelong immunosuppression after transplants. Regenerative medicine kind of aims to bypass a lot of that. Plus, it seeks to fundamentally enhance our normal wound healing processes and ideally minimize that problematic scarring that affects so many patients.
Okay, so our mission today is to take a truly deep dive. We'll explore the current state of stem cell biology, get into the cutting edge of tissue engineering, and really dig into the nitty-gritty of the latest clinical applications. We want to help you, the listener, understand exactly what's needed to bring regenerative medicine from the lab bench into routine clinical practice. Really, to help you grasp this information and its profound implications, like the elite professionals working in this field. All right, so at the absolute heart of regenerative medicine, the very foundation, really, are stem cells. We all hear the term stem cell thrown around quite a bit in the news and discussions. But what makes them so profoundly special? What is it about these cells that gives them this, well, almost magical potential to heal and rebuild?
That's a crucial question, absolutely. And it's what distinguishes them from, pretty much every other cell type in your body. Fundamentally, stem cells are undifferentiated cells. Think of them as like blank canvases. They haven't committed to a specific job yet, like becoming a skin cell or a muscle cell or a neuron. What gives them their unique power are two distinct and essential capacities. First, they possess what we call long-term self-renewal. This means they can divide and produce more stem cells. They essentially perpetuate their own population indefinitely, or at least for very long periods in the lab. They don't just divide and die off, they maintain their stemness, ensuring a continuous supply. Second, and maybe the most exciting part for regenerative medicine, they have the ability to differentiate. Under the right conditions, give them the correct biochemical signals, the right mechanical cues. A supportive environment, they can transform into multiple specialized cell types. This potential, to enter any desired differentiation program to basically become a lung cell or a heart cell or a bone cell, that's precisely why they're such a strong focus. They are, in essence, the body's master cells, just waiting for instructions.
So they're like a blank canvas, just waiting for the right instruction to become almost anything the body needs. That's a really interesting idea. The body has these uncommitted cells just waiting, ready for action. Given this incredible versatility, how are they usually classified? Based on this potential, right?
Precisely. That blank canvas analogy, it's pretty spot on. Traditionally, we classify them primarily by their differentiation potential, basically, by what they can become. The most versatile, the most powerful are pluripotent cells. These are the true powerhouses. They can differentiate into all three embryonic lineages. the ectoderm, mesoderm, and endoderm. Now, to put that into perspective for you, those three foundational layers, they give rise to every single cell type in the human body, your nervous tissue, muscles, internal organs, everything. So a pluripotent cell can literally form any cell type in the body. But, and this is an important distinction, they can't form extra embryonic tissue like the placenta. Think of it like this. A totipotent cell, which we'll get to, can build the entire house, foundations, walls, garden, everything. A pluripotent cell can build every single room inside that house, designing each one specifically, but not the garden or the outside support structures. Embryonic stem cells are the prime example here.
Okay,
Yeah.
got it. So pluripotent is any body cell, but not the support structures. What's next?
Next, we have multipotent cells. These are often called postnatal or tissue-specific stem cells. They're more limited. They can only give rise to specialized cells of a specific tissue type or lineage. Most adult stem cells fall right into this category. So, for instance, a blood stem cell. It can make all types of blood cells, red cells, white cells, platelets, but it typically can't just suddenly decide to become a neuron or a liver cell. Its fate is more restricted to its own tissue.
More specialized then. Are there others? You mentioned totipotent.
Yes, exactly. Beyond those two main groups, we have even broader or more restricted types. Totipotent cells, as I mentioned, are the most powerful of all, like the very first cells formed right after fertilization, a zygote, or a morula, that early cluster of cells. These can form all differentiated cells, and importantly, including those critical extra embryonic tissues like the placenta. So, in theory, one totipotent cell could form a complete organism.
Wow. Okay. And the other end of the spectrum.
Right. At the other end, we have unipotent cells. These are cells limited to producing just a single cell and tissue type. Think of cells like osteocytes, which are mature bone cells, or chondrocytes, mature cartilage cells. While they can divide and make more of themselves, their differentiation path is very narrow, strictly defined.
Okay, that gives us a fantastic foundation. We understand what stem cells are, how they're categorized by their potential. Now let's explore the different sources we can get these amazing cells from, the ones investigated for regenerative medicine. And we'll start with some of the most discussed and sometimes the most debated types. Hashtag tag 2.1, embryonic stem cells, ESC.
Right, embryonic stem cells, or ESCs, these are derived from what's called the inner cell mass of the blastocyst. Imagine a very early embryo, just a few days old before it implants in the uterus. It's this tiny sphere of cells, and inside is a small cluster that's the inner cell mass. What makes ESC so compelling, and sometimes, historically controversial, is they possess true pluripotency. They can differentiate into any somatic cell type in the body, full stop. Plus, they have an effectively unlimited capacity for self-renewal. You can grow them in culture, in lab dishes for very long periods, generating huge numbers of cells for research or potential therapy. Historically, when researchers first started working with ESCs, they often used animal material, like feeder layers of mouse fibroblasts. These provided growth factors to keep the ESCs undifferentiated. However, for human ESCs intended for clinical use, big advancements have been made. They're now meticulously cultured in what we call serum and feeder-free conditions. This gets rid of the risk of animal pathogens or unwanted immune triggers. And these human ESCs undergo extensive microbiologic testing, following strict recommendations from bodies like the International Stem Cell Banking Initiative. The US FDA also has incredibly strict rules: documentation of source, any genetic components, absence of pathogens, all aimed at ensuring the highest safety for patients.
Okay, so that's the what and how. Now, this is where the direct impact becomes clearer, their potential applications. What can these incredibly versatile cells actually do for disease and injury?
Exactly. The potential applications are vast, truly revolutionary on paper. Healthy human ESC lines are being established for large-scale cell banks globally. The vision is to have these banks ready to differentiate cells for reconstruction after, well, all sorts of debilitating conditions: congenital defects, trauma, infections that damage tissue, lesions from cancers. Beyond reconstruction, human ESCs are under intensive investigation for treating severe conditions like macular degeneration, a leading cause of blindness, various heart diseases, and even in cancer therapy itself. And they've been absolutely crucial for disease modeling. Researchers have derived ESC lines from embryos carrying known genetic diseases like Huntington's or cystic fibrosis or chromosomal abnormalities, these specific cell lines. They're invaluable tools in the lab. They let scientists study disease progression right there in a dish, giving a much better understanding of the causes, the pathophysiology, how the disease actually works. It paves the way for new drug discoveries.
That all sounds incredibly promising, like truly a medical revolution waiting to happen. But I know, despite this potential, there have been significant hurdles with ESCs. Scientific, societal, things that have slowed things down. What are those major challenges?
That's a crucial point. The path for ESCs hasn't been smooth, it's been complex. First, there are considerable technical, political, and ethical hurdles. Despite many established human ESC lines, you still see relatively few peer-reviewed publications on their clinical use, and a surprisingly limited number of actual clinical trials have been conducted. On the ethical front, blastomere harvesting, taking cells from an early embryo, raised significant concerns. The core issue for opponents is damaging potentially viable embryos. Now, techniques exist to isolate single cells without, supposedly, perturbing embryogenesis, but these haven't really satisfied opponents who fundamentally object to using human embryos. And what's truly profound here is these aren't just scientific debates. They touch society's deepest questions about the beginning of life. It makes their clinical journey uniquely difficult, a scientific marvel caught in a complex societal debate.
Okay, so the ethical and political side is complex. What about the science itself? Safety concerns?
Yes, significant safety concerns, particularly tumorigenesis, the risk of tumor formation. Their extensive pluripotentiality, combined with unlimited self-renewal, inherently means a risk of dysregulated growth if not perfectly controlled. That's a big one. What's more, the actual process of growing them in lab dishes, the in vitro expansion, it can subtly change their cellular and genetic biology, potentially predisposing them to form tumors once transplanted.
And immune response. Do they get rejected?
That's the other piece. While undifferentiated ESCs generally don't trigger an immune response, that changes significantly once they start differentiating. As they become specialized cells, they begin expressing major histocompatibility complex, MHC types 1 and 2. These are the flags the immune system recognizes. So, for clinical use, you'd need careful MHC matching, very similar to organ transplants, to avoid rejection by the patient's body. Another layer of complexity, hashtag 2.2 somatic cell nuclear transfer, SCNT.
Right, let's talk about somatic cell nuclear transfer, SCNT. Many of us immediately think of Dolly the sheep, right? Cloning. What exactly is the process here, and what was its promise for regenerative medicine beyond just copying organisms?
SCNT is fascinating, a really sophisticated technique. Fundamentally, it involves transferring a nucleus from a differentiated somatic cell, basically any body cell with the desired genetics, into an enucleated ovum, an egg cell with its own nucleus removed. The resulting reconstructed cell then divides, hopefully generating a blastocyst. And that blastocyst could, theoretically, yield a complete organism. Dolly the sheep, cloned back in 1997, was the first mammal successfully created this way. A huge scientific milestone captured everyone's imagination. Since then, over 20 mammalian species, including monkeys, have been cloned. And human embryos have been generated this way for research after optimizing the protocols. The potential of SCNT for human therapies lies in generating genetically matched stem cell lines. Imagine creating patient-specific stem cells. Because they share the patient's exact nuclear DNA, they theoretically wouldn't be rejected. This has huge implications for screening treatments, testing drugs on a patient's own cells, and, crucially, as a source of perfectly matched replacement cells for damaged organs, potentially eliminating the need for immunosuppression.
That sounds incredibly promising. Genetically matched cells, no immune rejection. Seems like the perfect answer. So why isn't this the standard approach? What are the real problems, the limitations that have stopped it from being widely used?
That's a perceptive question because, yeah, despite the theoretical promise, there are indeed numerous technical hurdles limiting SCNT's widespread use. First, a crucial issue. The resultant hybrid cells are often imperfect copies. The reprogramming process, where the egg's cytoplasm tries to reset the adult nucleus, isn't always complete or precise. This imperfection can lead to abnormalities, often seen in extraembryonic tissues, and sometimes in the cloned animals themselves if they develop.
Okay, so they're not perfect clones. What else?
Second, the cloning efficiency remains extremely low across all species. You need hundreds, maybe thousands of attempts, thousands of egg cells to get just one successful outcome. It's incredibly resource-intensive, expensive, and inefficient.
Right. That sounds like a major practical barrier.
Absolutely. And third, there's a problem with mitochondrial DNA. The enucleated egg, even without its nucleus, still has its own mitochondrial DNA, mtDNA, from the egg donor. This mtDNA is genetically distinct from the nucleus donor's mtDNA, so you have this mismatch between the patient's nuclear DNA and the egg donor's mitochondrial DNA. This itself risks immune rejection after transplantation, undermining that key theoretical advantage of perfect genetic matching.
So even the matching isn't perfect. Anything else?
And finally, a very practical limitation. There's a severe scarcity of high-quality donor-mature human oocytes available for research. You need these eggs to start the process, and their limited availability is a major bottleneck. All these technological and resource limitations, they really need to be overcome before SCNT could be widely applied clinically. It's ambitious science, but faces huge practical challenges. #tag 2.3 induced pluripotent stem cells, iPSCs.
Okay, this is where the story gets really exciting, I think, because it seems to sidestep some of those huge ethical and practical issues we just talked about with ESCs and SCNT. Tell us about induced pluripotent stem cells, iPSCs. This felt like a genuine paradigm-shifting breakthrough.
Oh, absolutely. When we connect this to the bigger picture, the discovery of iPSCs, it was, without doubt, a monumental breakthrough that fundamentally shifted the landscape. In 2006, Shinya Yamanaka and Kazutoshi Takahashi work that earned them a Nobel Prize made this landmark discovery. They found a set of just 4 specific transcription factors: Octatia4, SOX2, KLF4, and c-MyFic, the Yamanaka factors. What they showed was that by introducing these factors, they could effectively reprogram adult mouse somatic cells, like skin fibroblasts, back into cells that were remarkably similar to embryonic stem cells. The first iPSCs. The genius here isn't just bypassing ethical issues, but the revolutionary idea of rewinding an adult cell's biological clock. Personalized medicine potential that was previously unimaginable.
And then human iPSCs followed quickly.
Very quickly. In 2007, the first human iPSCs were generated from human fibroblasts. A truly pivotal moment. iPSCs immediately sparked immense interest because, yes, the ease and reproducibility of making them, and critically, the relative lack of those intense ethical or political concerns surrounding ESCs. One huge advantage is creating autologous iPSCs derived directly from the patient. Theoretically, this could drastically reduce or even eliminate the need for lifelong immunosuppression after transplantation, a major hurdle. Some studies even suggest iPSCs might be more tolerance-inducing than other transplanted cells, potentially allowing for extensive cell banking and using allogeneic cells from a donor without long-term immune suppression. That would be a massive step for accessibility.
So they're easier to make, fewer ethical issues, potential for personalized therapy. What are they being used for now?
Since 2007, the technology has evolved incredibly fast. Human iPSCs are now routinely used to model a huge variety of human diseases right in a dish. It's like creating a disease in a lab. Researchers can study progression, test hypotheses in a patient-specific context. They're being developed extensively for cell therapies replacing damaged cells. And they're invaluable for drug discovery, allowing high-throughput screening much more efficiently. Plus, CRISPR-Cas9 gene editing, that's further advanced iPSC modeling. Scientists can precisely introduce disease mutations into healthy iPSCs, or conversely, correct mutations in patient iPSCs, creating even more precise, tailor-made models.
This really does sound like the ideal solution, overcoming so many hurdles. It's easy to get swept away by the promise. But where do the practical challenges start to bite? What are the current roadblocks for getting iPSCs into widespread clinical use?
Right. While incredibly promising, translating human iPSCs from the lab to the clinic hasn't been straightforward. Significant hurdles remain. The primary concern, just like with ESCs, is the risk of tumorigenicity, that inherent pluripotency and self-renewal. It carries the risk of uncontrolled growth, if not perfectly managed. Making sure they don't misbehave in the body is paramount. This risk is so real that genetic mutations found in iPSCs actually led to the suspension of the very first human iPSC clinical trial back in 2014, a stark reminder about safety.
So tumor risk is still a major factor. Any other genetic issues?
Related to that, yes. iPSCs, much like ESCs, when grown in culture for long periods, are at increased risk of accumulating karyotypic abnormalities, changes in their chromosomes, and losing heterozygosity, basically genetic instability. These instabilities can contribute to that tumorigenicity risk. So because of these critical safety issues, rigorous methods to test and purify iPSC-derived products are areas of very active, intense investigation. Researchers are also working on robust ways to monitor for tumor formation after transplantation. The continuous, overarching challenge is preserving their self-renewal and pluripotency, keeping them useful, while simultaneously eliminating the tumor risk and, crucially, precisely directing their fate in vivo so they become only the desired cell type. It's a delicate, ongoing balancing act. #tag 2.4 fetal stem cells.
Okay, and what about fetal stem cells? We hear about them less often than ESCs or iPSCs. What makes them unique, and what's their current status in regenerative medicine?
Fetal stem cells are primitive cells derived from various fetal sources: fetal blood, liver, bone marrow, also amniotic fluid and placental tissue. What's notable is they share similar immunogenicity to ESCs, so they might trigger an immune response. But they're generally more restricted in their differentiation fates than ESCs, more multipotent than pluripotent. Under the right lab conditions, they can differentiate towards fat cells, adipogenic, bone cells, osteogenic, and cartilage cells, chondrogenic. However, a significant ethical debate surrounds their collection and use. Collecting fetal blood in utero, for example, carries risks of harming or terminating a pregnancy. That's a very serious concern. Currently, they're typically obtained from terminated fetuses, from tissues that would otherwise be discarded, which raises its own set of ethical considerations. So due to these profound ethical complexities and the practical challenges, fetal stem cells are, well, unlikely to become a routine source for general regenerative therapies anytime soon. That said, they might still provide a means for devising future autogenous in utero therapies treating certain conditions before birth, perhaps. A more specialized application. Hashtag 2.5 adult stem cells.
Let's shift then to adult stem cells, which I understand are maybe the most immediately relevant, given some are already in clinical use and have fewer ethical complexities. What defines them? How do they differ?
Right. In adult humans and many organisms, the inherent regenerative capacity of tissues is maintained by these tissue-specific stem cells, or adult stem cells. As we touched on, they are multipotent, meaning they can differentiate into some, but not all, tissue lineages, usually limited to cells found in their tissue of origin. A key characteristic is their differentiation is heavily influenced by their immediate microenvironment, the stem cell niche, which gives them crucial signals. Now, despite having more limited differentiation potential compared to pluripotent cells, adult stem cells offer that significant advantage. They can be isolated without the profound ethical concerns tied to ESCs or fetal stem cells. This ethical clarity has been a major factor solidifying their relevance and accelerating their progress towards clinical applications. It's often a more straightforward path. Historically, hematopoietic stem cells, HSCs, blood stem cells, were the first adult stem cells definitively described decades ago in mice. They've served as a foundational paradigm for studying all adult stem cell biology. More recently, mesenchymal stem stromal cells, MSCs, adipose-derived stem stromal cells, ASCs, and skeletal stem cells, SSCs, have become huge areas of research, and it's highly likely, as we learn more, we'll discover many more tissue-resident stem cells across various organs, uncovering more of our body's hidden repair mechanisms.
Okay, so let's break those down, starting with the first discovered, HSCs. Hashtag 2.5.1, hematopoietic stem cells, HSCs.
Right, hematopoietic stem cells, HSCs. They are arguably the most studied and best characterized adult multipotent stem cells we know. Their definitive isolation in mice decades ago was a breakthrough. These are the blood-forming cells. They reside in specialized niches, primarily in adult bone marrow. Their critical job is maintaining homeostasis of all hematopoietic cell lineages throughout life, continuously replenishing our red blood cells, white blood cells, platelets, and the clinical success story of HSCs. It's profound. Transplantation of HSCs for various blood diseases, leukemias, lymphomas, genetic disorders is, to date, the most widely used and successful stem cell therapy in medicine. It's been used for over 50 years. When these HSCs are transplanted into patients whose bone marrow niches have been cleared, say, by chemo or radiation to kill cancer cells, they have this remarkable ability to engraft, take root, and fully repopulate the entire hematopoietic system, restoring healthy blood and immunity. It really shows the power of cell therapy.
But there's a catch, right? You mentioned toxicity earlier.
Exactly. There's a significant limitation that curtails its broader use, especially for non-malignant conditions. The myeloablative regimens, those intensive treatments like high-dose chemo or radiation needed to ensure the transplanted HSCs engraft and survive, are highly toxic. They essentially wipe out the patient's existing marrow. This leads to severe side effects, high risk of complications. Because of this high toxicity, using HSCs for non-malignant patients where the disease isn't immediately life-threatening like aggressive cancer is currently considered unjustified. The risks often outweigh the benefits. Research is ongoing to reduce this toxicity, though. Hashtag 2.5.2 mesenchymal stem stromal cells, MSCs.
Okay, moving on to MSCs. These seem like a broad and maybe slightly confusing category. What exactly are mesenchymal stem stromal cells and what makes them so attractive?
You're absolutely right. MSCs are a broad and, sometimes ambiguously defined collection that's part of their complexity. They represent non-hematopoietic progenitor cells of mesodermal origin coming from that embryonic layer that gives rise to connective tissues, bones, muscles, structural components, basically. In the lab, they have characteristic spindle shapes, and they can form colonies from single cells. A significant advantage for therapy is they're considered immune privileged. This means they tend to evade or dampen the immune response when transplanted, a huge benefit, reducing rejection risk and the need for immunosuppressants.
And what can they become? What tissues can they form?
In terms of differentiation, MSCs can generate various connective tissue cells: osteoblasts for bone, adipocytes for fat, chondroblasts for cartilage, fibroblasts for connective tissue repair, and parasites around blood vessels. This range makes them attractive for therapies in orthopedics, wound healing, even cardiovascular repair. Importantly, MSCs are thought to mediate the regenerative effects not just by directly replacing cells. They seem to operate largely through paracrine signaling. Basically, they talk to nearby cells by releasing bioactive molecules, immunomodulators, antioxidants, anti-apoptotics, angiogenic factors, chemotactic agents. They create a more regenerative, healing environment, maybe more than becoming the new cells themselves.
That's fascinating, like conductors of healing, not just bricks. But you mentioned inconsistencies earlier, challenges with defining and isolating them. How does that impact things?
That's a critical point about standardization. There have indeed been substantial inconsistencies in the precise definition of MSCs, and crucially, in the methods used for isolating them. Because there aren't universally defined unique surface markers like a clear barcode, they've typically been isolated by a rather non-specific trait: their ability to stick to plastic culture dishes. This method, while practical, yields a heterogeneous population. The MSCs from one lab might not be exactly the same as from another, making study comparisons difficult. Developing standardized definitions and robust isolation techniques is absolutely essential for reliable clinical use. Because of this heterogeneity, MSC is increasingly used as an umbrella term for many tissue-specific multipotent stem cells, each perhaps with unique abilities. Understanding this mix is key to mastering their application. Hashtag 2.5.3 bone marrow-derived stromal cells, BMSCs. Right, so digging into that umbrella, bone marrow-derived stromal cells, or BMSCs, were among the first MSCs isolated back in the 1970s from bone marrow. They're actually quite rare, maybe 1 in 10,000 cells in the stromal fraction of adult bone marrow. They've been a strong research focus given their potential to form bone, cartilage, fat, and their immune effects. However, getting them involves a relatively invasive bone marrow aspirate, which isn't ideal for the patient. Plus, BMSCs often have low proliferation potential in vitro, meaning they don't expand into large numbers easily in the lab. This limits the therapeutic dose you can get. These factors combined make BMSCs maybe less attractive for widespread clinical use compared to other sources, which logically led researchers to look elsewhere for MSCs. Hashtag 2.4 adipose tissue-derived stem stromal cells, ASCs.
So if BMSCs are hard to get in large amounts, where else are MSCs easier to find? I hear fat tissue, adipose tissue is surprisingly good source. Seems a bit counterintuitive, is that right?
Indeed, absolutely true. It highlights the hidden potential in tissues we might just overlook. Adipose-derived stem stromal cells, ASCs, are located within the stromal vascular fraction of fat tissue. They've become incredibly attractive candidates precisely because they're abundant, and you can harvest them relatively easily through less invasive procedures like liposuction, much more patient-friendly and scalable.
Okay, easier to get. What can they do? What's their potential?
In vitro, ASCs can differentiate into bone, fat, cartilage, even muscle. Crucially, they also show potent in vivo effects: angiogenic, promoting blood vessel formation; adipogenic, fat cell formation; antifibrotic, reducing scarring; and anti-apoptotic, preventing cell death. This multifaceted capability makes them very promising for wound healing, reconstructive surgery, and more. Recent research suggests ASCs are actually a heterogeneous mix, multiple stem cell types, maybe even some pluripotent ones, all in that stromal fraction. But these individual cell types within the ASC population, they still need more detailed characterization to fully understand their specific roles and maximize their use. Hashtag 2.5.5 endothelial progenitor cells, EPCs. Okay, next up, endothelial progenitor cells, or EPCs, another intriguing cell population found circulating in peripheral blood. These cells express both hematopoietic and endothelial surface markers, suggesting a link between blood-forming and vessel-forming cells. Evidence suggests EPCs enter circulation in response to triggers like vascular injury or ischemia, lack of blood supply. Once mobilized, they're thought to actively participate in vasculogenesis, forming new blood vessels crucial for repair and getting oxygen to tissues.
So they help rebuild blood vessels. But you mentioned definition issues earlier, like with MSCs.
Exactly, a similar challenge. While generally thought to be mesenchymal in origin, there are no widely agreed upon isolation methods or defining surface antigens for precise prospective isolation. This methodological heterogeneity makes comparing studies and drawing firm conclusions very difficult, a common hurdle in developing fields. Recent research has tried to clarify by subcategorizing EPCs into at least two types. First, myeloid angiogenic cells. Hematopoietic-derived and, despite the name, unable to differentiate into true endothelial cells. They likely contribute indirectly, maybe by releasing growth factors. Second, and maybe more relevant for direct vessel formation, are endothelial colony-forming cells. These can self-assemble into functional blood vessels and express specific markers, VEGF receptor 2, CD146, CD31, E-cadherin, VWF, like unique barcodes, helping us identify and study them. This subcategorization is a step towards better understanding. Hashtag 2.5.6 skeletal stem cells, SSCS. Moving to the skeleton. A distinct population of postnatal skeletal stem cells, SSCs, has been identified in both mice and humans. These are true self-renewing cells, ensuring a supply of bone builders. Interestingly, they get actively upregulated after a fracture, showing their natural role in bone repair. They can differentiate into bone, cartilage, and bone marrow stroma, the supportive tissue. Importantly, they do not differentiate into fat, distinguishing them from some other MSC-like cells and making them specific for skeletal repair.
And we know how to trigger them specifically.
We're learning. The discovery that specific combinations of niche factors, those local biochemical and physical cues, can activate SSC programs in situ, right where they are, to form cartilage or bone. That has huge implications. These discoveries hold enormous promise for future cell therapies for skeletal disorders, osteosarcoma, bone cancer, complex fractures that don't heal well, damaged cartilage, which is notoriously hard to repair naturally. In humans, HSCs are specifically identified by three key surface antigens: CD73, CD164, and PDPN, podoplanin. This precise ID allows more targeted research and potential therapeutic manipulation, closer to tailored therapies for bone and joints. Hashtag 2.5.7 miscellaneous adult stem cells.
Wow, it really sounds like stem cells are everywhere, even in places we didn't expect, tissues once thought to have limited repair capacity. What other unexpected locations are researchers finding these resident stem cell pools?
This really highlights the joy of discovery, how our understanding keeps evolving. In recent years, yes, resident pools of tissue-specific stem cells have been identified in other organ systems, especially those with high cell turnover and significant natural regenerative capacity. Take the skin. Epidermal stem cells reside in niches along hair follicles, the bulge region, and in the deep interfollicular epidermis. These multipotent cells maintain normal skin homeostasis and regenerate it after trauma, crucial for wound healing. In the small intestine, highly proliferative cells at the base of the crypts, those are stem cells, they give rise to differentiated cells that migrate up, constantly repopulating the gut lining every few days. Incredible turnover.
And the really unexpected places: heart, brain.
Yes, perhaps most surprisingly, adult stem cells have also been isolated from systems previously thought to have little or no regenerative capacity, including cardiac tissue, heart, muscle tissue, and even neural tissue, brain and nervous system. This significantly shifted our understanding. These organs aren't as static as we thought. However, while these cells have been isolated, their precise role in vivo in maintaining local tissue homeostasis and contributing to organ regeneration, that's still under very active investigation. But their discovery opens entirely new avenues for therapies in heart failure, neurodegenerative diseases, pushing boundaries.
Okay, with all this talk of incredible growth, self-renewal, repair potential, it naturally raises the question: how do stem cells, especially when things go wrong, when the regulation breaks down, relate to diseases like cancer?
That's a crucial question, absolutely. It's a critical intersection between regenerative medicine and oncology. Mutations and dysregulation of our endogenous stem cells, the ones naturally in our bodies, are widely thought to underlie cancer biology. Many theories propose that cancer cells possess stem-like properties, often called cancer stem cells. These contribute to uncontrolled growth, metastasis, resistance to therapy. Therefore, as stem cell-based therapies move closer to widespread clinical use, it's absolutely critical to prioritize minimizing the risk of potential stem cell dysregulation and, importantly, the malignant transformation of the therapeutic stem cells we introduce. Ensuring the safety, long-term stability, and appropriate behavior of these transplanted cells is paramount. We have to avoid inadvertently causing new problems. This is a primary safety hurdle for all stem cell therapies.
So it's clearly not just about the cells themselves, the biological building blocks. It's also about how we deliver them, keep them alive, and crucially, control their behavior once they're in the body. That seems to be where the incredible field of bioengineering comes in, creating the right environment, the perfect home.
Absolutely. Understanding the complex biochemical and biophysical factors that contribute to success, it's essential for widespread clinical application. Stem cells are exquisitely sensitive to their surroundings, their niche. And this niche isn't just other cells and chemical signals. It also includes critical biophysical processes. Things like mechanotransduction, how cells sense and respond to physical forces, like stretch or compression, even electrical fields, temperature gradients. These specific factors likely play a crucial role in biasing differentiation fate, nudging a stem cell towards becoming bone versus cartilage, for instance. The required precision is immense.
So engineers are trying to recreate these niches.
Exactly. Substantial progress has been made in engineering what we call biomimetic microenvironments, engineered environments designed to mimic natural conditions in the body, capable of supporting engraftment, survival, therapeutic function, and helping direct stem cell fate and integration. There's even an exciting emerging field called regenerative rehabilitation. This combines regenerative medicine principles, like cell transplants, with traditional rehab, like physiotherapy or exercise. The goal is to enhance the success of these advanced therapies by training the body to integrate and use the new cells or tissues effectively. Like teaching a limb to regain strength, it acknowledges just implanting cells isn't enough. The body needs guidance to functionalize them. Hashtag 4.1 biomaterials as constructs for cell delivery and cell differentiation. Right, so a key tool here is biomaterials. These are designed as synthetic scaffolds that facilitate several key things: stem cell survival, engraftment into host tissue, proliferation, multiplying, and retention at the site of repair. Think of them as custom-built temporary homes or guiding matrices.
What are these scaffolds made of typically?
Commonly used ones include biocompatible materials like collagen polymers, natural structural proteins, polyglycolic acid, PGA, polylactic coglycolic acid, PLGA, and polyethylene glycol hydrogels. These materials are often chosen because they're porous, which is critical. Allows cells to get in, nutrients to reach them, waste to get out. They can also be easily molded and shaped sheets, tubes, complex 3D structures to fit the specific tissue need. Particularly innovative advance here is three-dimensional 3D bioprinting. This allows creating incredibly versatile scaffolds with precisely defined biomimetic properties closely resembling natural tissue structures and functions. These can provide intricate support, guide cell organization, even deliver growth factors exactly where needed microscopically. This tech is really helping bridge the gap between artificial constructs and complex native tissues.
Sounds amazing. But are there still challenges in making these scaffolds work perfectly in the body?
Oh, absolutely. While better delivery systems theoretically mean implanting fewer cells, there are still considerable challenges. Developing stem cell-specific delivery systems that truly mimic the physiological extracellular matrix of native tissue and, crucially, promote directed proliferation and differentiation in vivo, that requires incredibly deep understanding. We need to unravel complex cell-cell and cell matrix interactions and how physical stimuli in the dynamic body environment modify them. This research is still relatively early stage, pushing boundaries of biology and engineering. But future developments hold enormous promise for significantly boosting stem cell therapies, potentially leading to fully functional tissue restoration. Hashtag 4.2 organ-level tissue engineering.
Okay, this sounds like we're moving beyond just repairing tissues to actually building entirely new functional organs from scratch.
Yeah.
How close are we to that? Like printing a new liver or kidney that could actually work in a person?
Organ-level engineering is indeed the ambitious long-term goal, using advanced biomaterials and tissue engineering to create synthetic or partially engineered organs for transplant. It's a profoundly promising long-term solution to the persistent, critical shortage of donor organs. The vision is to eliminate waiting lists.
Where have we seen the most progress so far?
We've seen faster progress with hollow structures, which are simpler designs than solid organs. Things like blood vessels, urethral structures. They've reached clinical trials relatively quickly. For example, bioengineered human cellular vessels, basically scaffolds, seated with a patient's own cells, provided safe, functional hemodialysis access in phase 2 studies. A significant step for kidney disease patients.
But even simple structures have issues.
Yes, even with seemingly simpler hollow structures, challenges remain. There was an early success using an autologous cell-seated scaffold for bladder augmentation in spina bifida patients. Initially promising. But a more recent phase 2 trial showed it didn't actually improve bladder compliance or capacity long-term. Most individuals needed more surgeries due to complications like bladder rupture, bowel obstruction. This highlights a crucial point. Diseased host tissues might not provide a good environment for transplanted cells, even if the construct is sound. It often requires substantial optimization of both the construct and the recipient's biology for real long-term success.
Okay, so hollow structures are tricky. What about complex solid organs like liver or kidney?
Right, engineering complex, solid 3D organs, the challenges are exponentially greater. Additional cellular and material needs, precise tissue maturation over time, achieving complex, multifaceted functionality. Progress here has understandably lagged behind hollow structures. But despite the immense complexity, recent preclinical advances in bioengineering technologies have been truly impressive. Significant strides are being made, even if routine clinical use is still some way off. Researchers are getting closer, piece by piece.
And I heard about organoids, miniature organs in a dish.
Yes. A particularly exciting development under this umbrella is organoids. These are 3D in vitro culture systems derived from stem cells. They can remarkably recapitulate the architecture, function, and genetic identity of many solid organs. We have organoids for brain, liver, kidney, stomach, pancreas, ovary, intestine, skin, lung, a whole range. These miniature functional organ models are proving incredibly valuable for modeling diseases outside a living organism, for screening drugs much more efficiently than animal models, and for simulating regenerative therapies before human trials, even more transformative. Patient-derived organoids grown from a patient's own cells. This offers tremendous potential for personalized therapy, tailor-made drug testing, treatment strategies. Truly individualized medicine is getting closer.
Okay, we've covered the fascinating science, the different cells, the groundbreaking engineering. Now let's bring it all to life. Let's talk real-world clinical applications, the concrete results we're seeing from actual trials. Where is regenerative medicine truly making an impact today, and what are we learning from these early human experiments?
Right, this is where the rubber meets the road. Stem cell-mediated therapies have begun to transform the field, moving from theory into practice. A number are currently under careful scrutiny in rigorous clinical trials, evaluating safety and efficacy across a range of diseases. Hashtag 5.1, embryonic stem cells, ESCs, in the clinic. Okay, regarding embryonic stem cells, one notable clinical application was a phase I trial engineering monolayers, single layers of human ESC-derived retinal pigment epithelium, RPE cells. These RPE cells support the retina, crucial for vision. When delivered to this retinal space of patients with severe age-related macular degeneration, ARMD, the results showed improved visual acuity, a significant early sign of potential for treating degenerative blindness. Beyond that, ESCs are also being rigorously tested for generating insulin-producing pancreatic cells. The goal, to potentially correct type 1 diabetes, offering a curative solution by replacing the deficient cells. Hashtag 5.2 somatic cell nuclear transfer, SCNT, in the clinic. In the SCNT realm, a landmark study showcased its potential, giving a glimpse of what might be possible. Researchers combined the nucleus from a type 1 diabetic patient with a donated enucleated oocyte. The aim was to produce patient-matched stem cells capable of differentiating into those insulin-producing beta pancreatic cells destroyed in diabetes. While this showed immense promise generating patient-specific functional cells, significant regulatory hurdles, the procedure's complexity and funding issues have unfortunately hindered further translation into broader clinical use. The path from proof of concept to therapy remains incredibly challenging for SCNT. Hashtag 5.3, induced pluripotent stem cells, iPSCs, in the clinic.
What about iPSCs in actual human trials? This seemed like the most promising path, fewer ethical hurdles. Personalized medicine promise. What have the initial clinical results shown? What have we learned?
You're right to highlight iPSCs as a major area of hope. Their journey into the clinic has been groundbreaking yet instructional. The very first clinical study using human iPSC products launched in 2013, a huge milestone. It involved transplanting autologous iPSC-derived RPE cells reprogrammed from the patient's own skin cells into eyes damaged by ARMD. Initial reports from the first patient, incredibly encouraging. They received their own cells, showed no signs of immune rejection or tumor formation without needing immunosuppressants, a huge safety win, validating the personalized approach. At the one-year follow-up, visual acuity neither significantly improved nor declined. Stabilization, which for a degenerative disease is arguably a positive outcome.
But you said instructional. What went wrong?
While the path wasn't entirely smooth, unfortunately, the trial for the second patient in the study was halted. The reason given was the discovery of undisclosed genetic mutations found in the iPSCs intended for that second patient. This incident underscored that ongoing challenge of genetic instability with iPSCs and the critical need for rigorous, maybe even unprecedented, quality control and safety assessments before widespread use. While incredibly promising, it was a potent reminder the field is still actively developing, constantly refining protocols for safety and efficacy. It's that paradox. The versatility that makes them powerful also demands hypervigilance. Hashtag 5.4 bone marrow transplant, HSC transplantation, shifting to a therapy that has achieved routine clinical success. Bone marrow transplantation using HSCs, used successfully for over 50 years. It's a spectacular, enduring example of cell therapy's potential. It offers the best chance for cure for many malignant diseases, leukemias, lymphomas, and many non-malignant ones like genetic blood disorders, bone marrow failures. It also has the remarkable potential to induce tolerance in organ transplantation settings sometimes, helping a patient accept a donor organ without lifelong immunosuppression. Truly groundbreaking.
But we keep coming back to the toxicity.
Exactly. That significant limitation curtails broader use, especially for non-malignant conditions where the risk-benefit calculation is different. The myeloablative regimens, high-dose chemo, radiation needed to ensure engraftment, highly toxic. They wipe out the patient's marrow, leading to severe side effects, infection risk. Because of this, using HSCs for non-malignant patients often isn't considered justified currently. The risks can be life-threatening. Research continues on reducing this toxicity, though, perhaps with less aggressive conditioning.
Oh.
Hashtag 5.5 multipotent adult stem cells, MSCs, ASCs, EPCs, in the clinic.
So what's the latest with the other multipotent adult stem cells we discussed, MSCs, ASCs, EPCs in clinical trials? Are we seeing clearer, more consistent results here, given their advantages?
It's a mixed bag of results, honestly, highlighting the ongoing iterative nature of scientific development. We're seeing promise, but also areas needing significant further optimization and understanding. It's part of the rigorous process. For bone marrow-derived stromal cells, BMSCs, after encouraging preliminary results, autologous BMSCs were used to treat chronic advanced ischemic heart failure in a large phase 3 trial, a maybe step. However, the overall outcomes, no significant benefit in the treated group versus placebo up to 39 weeks. Interestingly, a post-hoc analysis, digging deeper into the data at 52 weeks, suggested a U-shaped response curve. Patients getting a medium dose showed the most benefit. Low or high doses were less effective. This isn't just quirky data. It tells us that even for cells in trials, precision dosing, like with drugs, is paramount and still being figured out. We need more nuanced studies to find that optimal therapeutic window.
Okay, so dose matters. What about ASCs, the fat-derived cells, for perianal fistulas?
Right. Adipose tissue-derived stem stromal cells, ASCs. Two key trials to compare. One phase 3 trial delivered autologous ASCs with or without fibrin glue into complex perianal fistulas in patients without inflammatory bowel disease. Results: no greater benefit compared to glue alone at 6 months. The cells didn't add significant value for this group. However, a different phase 3 trial looked at chronic fistulas associated with Crohn disease. Here, injecting 120 million allogeneic ASCs from a donor without glue was superior to placebo in closing fistulas. Benefits were seen at 24 weeks and lasted at least a year.
So same cell type, different results depending on the disease context and maybe using donor cells.
Exactly. It highlights a critical lesson. Context, dosage, maybe even donor versus patient cells, they all matter. It requires highly tailored approaches.
And MSCs for graft versus host disease, GVHD.
Mesenchymal stem stromal cells, MSCs, have also reached phase 3, mainly for their immunomodulatory properties, dampening immune attack. One trial for steroid-refractory GVHD showed no significant benefit over placebo at 28 days. Disappointing. But a second trial, focusing on a select pediatric group with severe steroid-resistant GVHD and strict criteria, it did find significant improvements in MSC-treated patients versus placebo at 28 days. This difference might mean patient selection, disease severity, timing of administration are critical for MSC efficacy. Plus, a recent phase I trial reported encouraging early results using iPSC-derived MSCs for adults with GVHD, a potential new avenue combining iPSC advantages with MSC properties.
And EPCs, the vessel-forming cells.
Endothelial progenitor cells, EPCs, have been used in numerous studies for vascular injury, trying to restore blood flow. But a significant challenge is the substantial heterogeneity in reported methodologies, different isolation techniques, cell numbers, delivery methods, even definitions. This makes it very hard to compare results and definitively establish consistent efficacy. The field needs standardization to get clear answers.
So lots still ongoing.
Absolutely. A number of other phase 3 trials are underway exploring MSCs for Crohn's, chronic heart failure, chronic back pain, more pediatric GVHD studies. These are mostly industry-sponsored, focused on demonstrating efficacy of banked, thawed allogeneic MSCs, if successful, using pre-prepared donor cells from a bank. That would greatly simplify logistics, improve accessibility, make therapies much more widely available. Hashtag tagged conclusion. So, if we connect all this back to the bigger picture, it's clear regenerative medicine and stem cell based therapies are profoundly promising. The potential to restore function, alleviate suffering, truly rebuild damaged tissues and organs, it's revolutionary, inspiring. However, their full clinical potential, as we've seen from those very trial results, well, it's yet to be fully and consistently realized. Consider this: a considerable amount of rigorous groundwork is still required. We need to consistently confirm the safety, the efficiency, the long-term effectiveness of engineered tissues and cells before widespread application becomes routine clinical practice globally. This constant tension, between the exhilarating speed of discovery and the meticulous, sometimes slow pace needed for absolute patient safety, that's the defining paradox here. Progress is immense, but it often feels like a carefully choreographed dance between innovation and validation. And this raises an important question for all of us scientists, clinicians, regulators. How do we effectively balance that incredible drive for breakthrough innovation with the absolute necessity for careful, meticulous validation and patient safety?
So what does this all mean for you, the learner, trying to master this incredibly complex, fast-moving field? Well, there are certainly challenges, careful steps ahead. It really sounds like the field is rapidly accelerating, making major strides year after year, constantly learning from both successes and, frankly, setbacks.
Exactly. The journey is complex, but the pace is exhilarating. Exciting advances in biomedical sciences, ingenious engineering approaches, they're significantly speeding up the whole process. It is highly likely that many important milestones, both in fundamental understanding and clinical application, will be met in the upcoming years. The journey from initial lab discovery to routine clinical practice. Yes, it's complex, filled with scientific and ethical considerations, but the potential to restore function, alleviate suffering, and fundamentally overcome current limitations in medicine, it truly is immense. It's a field actively shaping the future of human health right now.
So here's a thought to take with you, something to mull over, maybe fuel your own curiosity about this groundbreaking field. If our bodies, through evolution, possess this natural capacity for speed and strength of repair that often leads to scarring, imperfect healing, yet other complex organisms, like those salamanders, can regrow entire limbs, perfectly restore organs. What fundamental biological switches might be hidden within our own genetic code that regenerative medicine is aiming to rediscover, maybe reactivate? And how might that deeper understanding eventually redefine our very concept of healing, moving us beyond just repair to a future of true, comprehensive regeneration for humans? It's a profound thought, and one that continues to drive this incredible field forward.